Today we will talk about gram stain. A 10-day-old newborn was brought to the hospital with symptoms of fever, irritability, and poor feeding. A lumbar puncture was performed, and the cerebrospinal fluid was found to be cloudy with a high white blood cell count. Gram stain of the CSF revealed gram-positive cocci in pairs and chains. The newborn was diagnosed with meningitis and started on antibiotics. What is the most likely cause of the newborn meningitis based on gram stain result? A. E. coli, B. Neisseria meningitis, C. Listeria monocytogenes, D. Staphylococcus aureus, E. Streptococcus agalactiae, or group B. Streptococcus. Well, based on gram stain, we have gram-positive cocci in pairs and chains. Streptococcus agalactiae is the only choice in the question that is gram-positive cocci in pairs and chains. Therefore, choice E, Streptococcus agalactiae or group B Streptococcus is the correct answer. It is a significant cause of meningitis and pneumonia in newborns. What about other incorrect answers? Option A, E. coli, is a common cause of late-onset meningitis in newborns, typically occurring after the first week of life, but it is gram-negative rod. Option B, Neisseria meningitis, is a common cause of meningitis in older children and adults. It is a gram-negative diplococci. For choice C, Listeria monocytogenes, it can cause meningitis in newborns, but it is gram-positive rod. For option D, Staphylococcus aureus, can cause a variety of infection, but it is not a common cause of meningitis in infants. In addition, it is gram-positive cocci in clusters. Let's talk a little bit about the gram stain procedure. The correct order of the gram stain procedure is as follows: fixation, crystal violet, iodine, alcohol acetone, and safranin. What is the function of iodine? A. Primary stain, B. Mordant, C. Decolorizer, D. Counterstain, E. None of the above. So the correct answer is B. Mordant. Let's talk about steps of gram stain. First, we fix the bacteria on the slide. Then we add the primary stain, which is crystal violet, that enters the peptidoglycan of all bacteria, giving them a purple color. The next stain is gram iodine, which is the mordant, that combines with the crystal violet to make a bigger complex in the peptidoglycan wall. After that, ethanol is used as a decolorizer, which will remove the crystal violet iodine complex from thin peptidoglycan layers of a gram-negative cell wall, but not the thick peptidoglycan layers of a gram-positive cell walls. After the colorization step, gram-positive cells will still hold onto the purple crystal violet color, but gram-negative cells will lose the purple color and be transparent. We then add the counterstain safranin, which stains gram-negative cells as pink color. Which one of the following bacteria has poor staining by gram stain? A. E. coli, B. Streptococcus pyogenes, C. Staphylococcus aureus, D. Chlamydia trachomatis, E. Acinetobacter baumannii. Well, the correct answer is D. Chlamydia trachomatis, and that's because of the unique cell wall structure of Chlamydia trachomatis, and it is an example of bacteria that has poor staining with gram stain. What about the incorrect choices? E. coli and Acinetobacter baumannii are gram-negative bacteria that stain pink by gram stain, while Streptococcus pyogenes and Staphylococcus aureus are gram-positive bacteria that stain purple by gram stain. From this, we see that gram stain is one of the most widely used laboratory techniques in microbiology, and it allows us to differentiate bacterial species based on their cell wall structure, providing valuable information for diagnosis and treatment of infections.